Hello friends, how are you doing? And I hope you really are all doing good. So today we will discuss the genetics chapter, molecular basis of inheritance, the NEET pattern exercise, right? The first question, which of the following is not a part of the transcription unit in the DNA? Okay, transcription unit. So the moment you see the options, the inducer, terminator, promoter, structural gene, your idea goes to the lacopron. Okay, you remember we talked about the lacopron when we were discussing this chapter, right? Right, and this was what? Here was the promoter, the terminator, right, and here was the structural gene. The structural gene was, you remember, when we talked about the lacopron, there was ZYA, beta galactosidase and all those things, permease and all that, you remember? And so the inducer, the terminator is here, then the promoter is here, the structural gene is here. So what is remaining? What is remaining is the inducer. So inducer is, as you remember, the lacopron, we discussed what? One is, okay, before this we said that the opron can be of two types. Right, we said opron can be of two types, right? One is the lacopron and the other one, one is inducible opron and the other one will be the repressible opron. So number one will be the inducible opron and the other one will be the repressible opron, right. Now inducible opron, as the word says inducible, it will be inactive. I hope you also are active now if you were sleeping. Okay, so inducible opron is one which is inactive, and if you give a promote, if you give some stimulus to it, it will be induced, and that is why the word inducible opron. However, when we talk about the repressible opron, this will be in the active state, and it has to be repressed, and that is why we say repressible opron. So inducible opron will be induced by an inducer, right? This opron will be induced by an inducer, right? This opron will be induced by an inducer, and that is what an inducer is. This inducer will come and bind here. It'll come and bind here, and it will activate the whole of the opron. In case of when we're talking about the lacopron, it was the lactose and the allolactose. Okay, the lacopron was basically for the digestion of the lactose in the prokaryotes. So the inducer will be the option here, which is not in the transcription unit, right? So we move on to the next question. If one strand of DNA right. So if one strand of DNA has a nitrogenous base sequence as ATCTG, what would be the complementary strand sequence? C. Whenever we're talking about the nitrogenous bases, one thing is the amount of purines will be always equal to the amount of pyrimidines, right? the amount of purines or purines the amount of purines will always be equal to the amount of pyrimidines, right? Even the, even the sound of the chalk, seems to make you laugh. Good, you should keep laugh. That means the adenine will be equal to thymine and the guanine will be equal to cytosine. Obviously, we know that here there will be 2 hydrogen bond and here there will be 3 hydrogen bond, right? 2 hydrogen bond and 3 hydrogen bond. So this is the purine and this is the pyrimidine. Those were the Chagaff's base pairing and all that. Remember Chagaff's base equivalence rule and all that when we discussed about these. So in that case, always adenine will pair with thymine and guanine will pair with cytosine in that case. And when we are talking about the RNA, this thymine will be replaced by the what? Uracil. In RNA, the thymine will be replaced by uracil. Okay. So in RNA the thymine will be replaced by uracil. So hence let us see ATCTG. ATCTG. Now let us see the complementary. Adenine is equal to what? Thymine. Okay, and this is replaced by what? U. Then thymine will be replaced by adenine, cytosine, guanine, thymine, adenine, guanine, cytosine. UAGAC, UAG. This is how you should pronounce UAGAC. Right, this is nothing, no word like that, just kidding. So this is your correct option. We now move on to the next question. What are the structures called that give an appearance as beads and string in the chromosomes when viewed, okay, when viewed under the electron microscope? Yes. The genes, the nucleotides, the nucleosomes and the base pairs. Once again the question, what are the structures called that give an appearance as beads and string appearance? Now this is talking about the chromatin. Okay, the chromatin will always appear as the beads and string appearance, and this is what called as the nucleosomes, right? This is what is called as the nucleosomes. Now what are nucleosomes? What we see in a nucleosome, what is happening over here basically, that the Packer molecules are there. Histone, nucleosome is actually a core of histone molecule which is wrapped by the DNA outside, and the core consists of what? The core consists of 4 histone molecule, H2A, H2B, H3 and H4. The core is known as Nu, NU. Right. So what we say, the nucleosome consists of an octamer. Why octamer? Because two molecules each of 4 histone molecules, that makes 4 histone molecules, right? So the nucleosome consists of an octamer consisting of 4 histone molecule type, 2 molecules each. The nucleosome consists of an octamer consisting of 4 histone molecule, 2 molecules each. That means H2A, H2B, H3 and H4. H2A, H2B, H3 and H4, this is what is the octamer, right? The DNA is wrapped around this octamer molecule. The DNA is wrapped around this octamer molecule. Okay, the DNA is wrapped around this octamer molecule. This DNA, the core of this is called as the nu. The core of nucleosome is called as nu, right? The core of nucleosome is called as nu, and further what we will see, that two nucleosomes are connected by what? By linker DNA, which is associated with what? Which is associated with H1 histone. So two, I think a little more in detail. Okay, this is last line I am giving you for this question. Okay, so two nucleosomes are connected. Two nucleosomes are connected by linker DNA, which has H1 histone molecule. Okay, so the answer will be nucleosomes. We move on to the next question. What is not true for genetic code? The genetic code was GAMO was the person responsible for the genetic code. A codon in mRNA is read in a non-contiguous fashion, not at all right. It is read in a continuous manner, contiguous fashion, the codon in the mRNA. What are the salient features of the genetic code which was given by GAMO? The salient features of the genetic code. Okay, the salient features of genetic code, right? It consists of triplet base sequences of the mRNA, right? Triplet sequences which are without the comma, that means comma less. Read in a continuous fashion, read in a continuous fashion, right? These are non-overlapping, not non-overlapping, non-overlap overlapping, then these are degenerate codes. Yes, unambiguous, non-ambiguous, degenerate, nearly universal, absolutely, but this is wrong, and hence this is the correct option. Okay, this wrong you can consider it another way. So the correct option will be here, the first one. Okay, we move on to the next question, the Okazaki fragments in DNA chain growth, right. So what we see here, okay, result in transcription, polymerize in the 3' to 5' direction and form the replication fork, prove semi-conservative nature of DNA replication, and polymerize in the 5' to 3' direction and explain the 3' to 5' direction. Now what are the Okazaki fragments? Right. So what is happening, this is, see, this is the double template DNA. We are talking about what? The Okazaki fragments in the DNA, polymerize in the 3' to 5' direction. So now we see this is the double stranded DNA. What happens, the double stranded DNA does not unwind completely. It unwinds, the double stranded DNA unwinds only up till a particular region and forms a Y-shaped structure. So if something is like this is the fifth question. If we open this, if you open the coil, how will it become? Won't it become like this? Yes, it will become like this. And that is why we say, it represents the replication fork, okay, the Y-shape. So this whole thing is actually, you can say, the Y-shaped replication fork. Okay, the Y-shaped replication fork. So what is happening? First of all, with the help of the enzyme, we'll do that. I think there's a question regarding that also. The DNA gyrase, the enzyme which will unwind the helix, which will open the helix up to a certain point, is the enzyme DNA gyrase, and then the two strands will be there. Now what is happening, in this strand, the replication is taking place in the direction of 3' to 5', and the replication will be in a continuous manner, and this strand is called as the leading strand. But in the other strand, that is in the direction of 3' to 5', the synthesis or the replication is in fragments, and these fragments are called as the Okazaki fragments, right? Okazaki fragments. And this strand is called as the lagging strand, and hence, based on this, we will see the different options. The Okazaki fragments in DNA chain growth results in transcription, polymerize in the 3' to 5' direction, 3' to 5' direction, and forms the replication fork. This whole thing is the replication fork. Yeah, that's what I've written here, the Y-shaped replication fork. So, this is the correct option. Okay, forms the replication fork. Next, let us move on to the next question. Which statement is correct for the bacterial transduction? Okay, the bacterial transfer is, we know that it takes place by transformation, transduction, as we know, as well as by conjugation. Transfer of some genes from one bacteria to another bacteria through the virus. Yes, this is the correct. What is happening in transduction? There is the transfer. Okay, what does basically transduction mean? The transfer of genetic material, transfer of genetic material from one bacteria to another by means of the virus. So, transfer of genetic material from one bacteria to another with help of virus. Okay, with the help of the virus, right? However, what was there, in the transduction? After I also told about your conjugation. Conjugation. In the conjugation, there was the formation of conjugation tube. There was the formation of conjugation tube, which was helping, right? There was the formation of conjugation tube, which was helping in the transfer of genetic material from one bacteria to the other. Okay, so conjugation takes place in the presence of conjugation tube, and so what will we see? The transfer of genes from one bacteria, transfer of some genes from one bacteria to another bacteria through the virus. Hence, this will be the correct option. Right. Let us move to the next question. The single hypothesis for the biosynthesis of secretory type of proteins was proposed by. Right. Now, the single hypothesis, what is happening, when the proteins are synthesized, okay, when the proteins are synthesized, they need to have a direction where to go. They need to be known, knowing where to go. So, what happened, there was a single amino acid hypothesis which said that there was, amino acid sequence at that particular site where the proteins were there, which will guide the protein, okay, which will guide the protein for its, for its where its direction, for its, its end where it has to reach, and that was given by Balbil and Sabatini. Right, the Balbil and Sabatini, the single hypothesis. Right. Let us talk about the next question. Protein helping, we discussed this question already. Protein helping in the opening of the double DNA helix, right? In the opening of the double DNA helix. So, what was it we said? Or rather, this we can say, right. So, this is the double helix, this is the double helix, and we see it has opened like the replication fork, and that is what we can say, the enzyme DNA gyrase, right. DNA gyrase helps in the opening, of the double DNA helix. Okay, so DNA guidase helps in the opening of the double DNA helix, and this is what we will see, the DNA guidase, right. We will talk about the next question. The nucleotide arrangement in the DNA can be seen by the what? Right. The nucleotide arrangement in DNA, okay, the 3D dimension was first in the X-ray crystallography, was first given by Astbury, right. The nucleotide arrangement in DNA, right, by the X-ray crystallography, by X-ray crystallography, okay, by X-ray crystallography was first given by Astbury. Okay, it was first given by Astbury. You remember, this X-ray, yes, the 3D structure of DNA was given by whom? That was, that the work was also done by Wilkins and Franklin, right, the Wilkins and Franklin and all those things. Okay, you remember even we talked about the semi-conservative, I think we are going to talk about that in the last, the last question, the semi-conservative nature of the DNA, right. So, X-ray crystallography. Now, we will talk about the last question. DNA replication is what? See, DNA always replicates in what way? Whenever we are talking about a strand of DNA, we all know DNA is double stranded, okay. DNA is double stranded. DNA is double stranded. So, what will always, what will happen, one of the strand will act as the parent strand, and there will be, the daughter strand, based complementary to that parent strand. So, always, always the semi-conservative nature. Why semi-conservative? Because in the new DNA form, one is the parent DNA, one is the parent strand and the other is a daughter strand. Similarly, here we will see here, one is the parent strand and the other one is the daughter strand. So, the semi-conservative nature, had it been both the same conservative, but how come possible, that would have been possible. So, the semi-conservative nature of the DNA, yes, the semi-conservative nature and also the semi-discontinuous nature. And who perform experiments to prove the semi-conservative nature of DNA replication? Michelson and Stahl. Okay, the Michelson and Stahl, they did experiments to prove this DNA replication is semi-conservative, right. So, friends, I hope you are following this and things are it is fine. We will do another set of questions in the near in the later sessions, but as of now we will just wrap it up here. Thanks a lot for watching and do stay healthy, and thank you. Bye bye.